More Bonani Tobela Dumela. So welcome to the SAIAB seminar series. Today's speaker definitely doesn't need an introduction. I think a lot of people are familiar with him. It's Dr. Tadiwa Mutizwa. He's a postdoc in the Freshwater Research Group under Professor Albert Chakona. Yeah, I'm still getting used to calling him Prof. So Tadiwa did his MSc and PhD through SAIAB under the supervision of Professor Chakona. He's a taxonomist by training, and having seen his progression over the years in terms of freshwater taxonomy, I can say he's not just one for the future, he's one for the present. And I think soon, Prof. Chakona, you will retire nicely. Tadiwa will carry on the baton. So Tadiwa will be giving us a talk on the diversity. I almost said ecology, being an ecologist, the diversity of Suckermouth catfishes of the genus Chiloglanis in Southern Africa, and in particular the Zambezi River. So those online, just don't be afraid to type questions in the chat. Prof. Mutizwa would really like to get some good input. And also you guys in the room. Yeah, it's a feature prof. So not to steal your thunder, ladies and gentlemen, give you Dr. Tadiwa Mutizwa.
Good morning again. Thank you, Mandla, for the introduction. The more he keeps talking, the more our accolades I kept getting. I started as Tadiwa, then I became doctor, and I was prof by the end. So I hope that continues. So yeah, without wasting a lot of time, SAIAB has been a place that I've been calling home for a very, very long time. So this is more going to be a casual talk rather than a formal conference type presentation. So yeah, let's get the show rolling. Okay, so first of all, we'll start with what is it that we do? What is it that I do? So I work with systematics and taxonomy, and what does that necessarily mean? So when we are talking about systematics, you're trying to talk about how you find relationships between organisms, how the different groups are related to each other. And some of the ways that we are able to investigate these kind of relationships are through genetic diversity, morphology, ecology, distribution. So when you have one species there and species A and species B, how are they related and how do they interact? And that forms part of systematics. Then the taxonomy part is more of the practical part where you are, in cases where you find unique organisms, you have to describe them, you have to give them names. And what is the importance of all this? So systematics answers the questions of what diversity do we have? How do you describe the diversity? How are the different groups related? And where do you find them? That's part of the taxonomy. And this is very important because now when you then go to the taxonomy, which then gives you this description part, it is important because accurate delimitation of species and taxa and different levels of biodiversity are important when you come to doing your research. So taxonomy essentially forms the basis of biological research. And it gives us a universal way of communicating ideas about the different life that we find on Earth. So, and the reason why I say universal, it's very important to always remember that once a species has been given a name, a species name, even though whether it gets revised or not, that name is applied globally. So if I name a species here and I publish that name, that name is going to be used globally. So all the species descriptions that we have a global impact, and they will remain in the record of that particular group of organisms throughout history. Because even if someone later comes on and is like, Tadiwa maybe made a mistake here, let's change it, you still have that record. And this is the common language that all biologists use. Manda was talking about being an ecologist, but when you talk about ecology, how do you study your ecology if you don't know what you're talking about? And this is where the important parts of what we do as taxonomists comes in, as well as elaborating the relationships of all these different organisms. And also, it's not just an academic exercise. It's also something that's enshrined within the South African Constitution. And there are provisions to ensure that we have institutes running in the country that ensure that we are aware of what biodiversity we have, where it is, what threats it faces, and even the whole act of actually designating species name. It's all included within the constitution of the country, and not just South Africa. It is also even a global pact. We even have international organizations that are dedicated to ensuring that we are able to quantify all the biodiversity that we have. And how does this then affect us? So this is just a small excerpt that I took from the National Biodiversity Assessment 2018, where biodiversity, okay, so I'll just read the excerpt. This biodiversity wealth gives our people tangible benefits like food, clean water, medicine, materials, and it supports agricultural and fisheries production and helps protect from natural hazards like floods, droughts, provides basis for vibrant tourism industry while offering natural spaces for recreational and activities. Biodiversity is central to South Africa's national objectives of addressing poverty, inequality, unemployment, and supports increased economic growth and improved service delivery to citizens. So for you to then get to these more higher goals, these more application kind of goals, you first need the fundamentals. How do you make a decision on conservation if you are not able to quantify the biodiversity? If you do not know what is in the system, what is in the ecosystem, and what role it plays in the ecosystem. So that is why our research comes in, forming that basis for all these other researchers. So yeah, now going into what we've been doing here as a group at SAIAB. SAIAB is essentially a leader on the continent when it comes to the taxonomy of aquatic biodiversity. You look at any taxa, any group, you always find either SAIAB scientists or HRAs, our associates, or people who came through the SAIAB system or associated with the SAIAB system. And some of the people are present here and are actually working on the taxonomy of freshwater fishes. So this is essentially the team, of course, under the leadership of the fearless leader, Albert. And of course, we have a lot of people from diverse, and you can see it's quite a diverse group, from people from, different age groups to different sexes, different people from different parts of the world, and we're all working towards a united goal. So examples of my colleagues' works, essentially it's not just a plug. So we have work from Dr. Peters. He's essentially the guy you go to when you have questions about barbs. And he's been working very hard to provide a lot of descriptions and descriptions of all these little barbs that we find in our water systems. South Africa this is probably one of the most diverse groups that you find within South Africa. And he is working towards resolving some of the taxonomy for that group. We have Dr. Stole sitting in the crowd as well. She's been also working a lot on the marine side, but she also dabbles with some freshwater stuff as well. And she's been working on these examples of her work, which is the catfishes, Chiloglanis, and some of the species that she recently described, as well as work from Dr. Zare Fatah. He is working very hard again. This guy publishes like crazy. A lot of work, again, working on pseudobarbers. And of course, he's very passionate about gobies. If you go to Fatah's office, if you just get stuck in the conversation about gobies, you're going to be there for an hour. So, of course, again, work that is done, describe several new species, including the famous one named after Voyani here. So, essentially, it's a very productive group. We are trying our best because there is a very big gap in the taxonomy of freshwater fishes, particularly in Africa. So we are trying to cover that gap so that when Mandla goes and does his fancy ecology stuff, he actually knows what he's talking about. So now into the actual talk. That was just introduction. So I am going to be talking about the Chiloglanis group, freshwater. These are small little tiny catfishes, they don't exceed more than, you hardly ever get anything more than 15 centimeters, usually typically associated with freshwater riffle systems, fast flowing waters, and let me take you into it. So they are part of the family Mochokidae, which is one of the most diverse groups in Africa. It's unique to Africa. The Mochokidae, you don't find them anywhere else except Africa. And Chiloglanis is probably one of the biggest groups, second only to Cynodontis. With the family has over 226 species. I think there are now more. And within Chiloglanis, we have more than 51 species. And they show a lot of diversity. They don't show a lot of diversity morphologically because they are very adapted to the environments that they live in, the fast-flowing waters. And one of the main features that you notice is this feature, the Suckermouth. So they use this to attach to surfaces as well as to feed, because they are usually found in fast-flowing waters. And this is maybe one of the major characteristics. When you pull out, you can tell this is a Suckermouth catfish. And these are examples of some that you find in South Africa. This is Anoteris, this is Paratus, and this is Swiestrai. So unfortunately, despite this group being very diverse, you find within Southern Africa, most of the diversity you find within the Central Africa, Congo, and West Africa. But within Southern Africa, we only have, I think there are now 10 species now, and 6 of them are found within South Africa. And you see this very small region, that's where you find 6 of them, which means the rest of Southern Africa, currently, we only have 4 species, which is very peculiar for taxa that are associated with very specific habitats. And this is something that raised some of our interest, and we started to look into it. And yeah, many people have also worked on it. I'm not the only one who's worked on this. So moving on, work by Chakona et al. 2018. He did a study with colleagues, and they found that within the Eastern Zimbabwean Highlands, they actually found 2 unique lineages, as well as other lineages that you find within the Zambezi River system. So already just from genetic studies, without even looking at morphology or even larger widespread, you can already find that there are actually a lot of hidden diversity within. Hence, we started this whole hidden diversity in series, if I may call it. So we moved on to more recently, we had another study by Day et al. They did a much more comprehensive sampling across all of Africa. And from their study, they were actually able to identify more than 20 unique lineages. And all of these things are undescribed. So you find that some species that are currently valid may contain 4, 5, 10 unique lineages that potentially represent species. And this is a case that we then went on to further investigate. So with work that we did in 2025, we described a new species from the middle Zambezi, Chiloglanis Peters. Yeah, one of my favorite species, was my first ever species description. Then we moved on. We went to the Eastern Zimbabwean Islands, following up on work that had already been set up by Prof. Chakona. We described 2 new species from the Eastern Zimbabwean Islands. So this small area, we were able to describe 2 new species, Asperacutis and Compactus. Then now, currently what we are working on is within the Zambezi River system. So within the Zambezi, as it is, we have a species, but we don't have a species. So what used to be in the Zambezi used to be called Chiloglanis pneumoni, but recent studies have shown that Chiloglanis pneumoni is actually an East African species. So that essentially orphaned the Zambezi Chiloglanis. So we know they are Chiloglanis, but they don't technically have a name. It's just Chiloglanis Zambezi as it is. So that is what I have been working on, and this is what I'm some of the results that I'm presenting for this talk. So this species was described, like I said, it should be Chiloglanis pneumoni, but we said recent studies have restricted that to Tanzania. So what we have in the Zambezi essentially is just a group of things that don't really have a name. So the aim of this study was to provide formal descriptions for undescribed species within Chiloglanis Zambezi species complex by applying an integrative taxonomic approach. So integrative taxonomic approach essentially just means you're using evidence from multiple sources, from the genetics to the morphology, and in some cases, not necessarily here, but in some cases, you can even use the ecology and all these other aspects. But in this case, we focused on the genetics as well as the morphology. So yeah, this is me measuring stuff. So I did measure stuff, guys. This is the key that I used. as well as X-rays that the collections people graciously provide for us, as well as, so moving on to the results within the Zambezi. So mind you, this is just only the Zambezi stuff. So all the red stuff are potentially new species with, of course, these are South African species that you have to add them so that you can have a property. So we have species from the Ruo, which is very interesting because you find in the Ruo, you have this one called SP Ruo, then you also have this one which I call SP Shield. These guys are not even sister taxa and they co-occur. So the patterns of diversity in there, when you start thinking about the biogeographic implications, like how would you have something that are in very different parts of the phylogenetic tree, and yet they co-occur? How did that occur? Then, of course, you will have some closely related, like the Lower Zambezi has quite, I suspect a lot of the reason why we have a lot of representation is because we have quite a lot of tissues from that region. But if we were to comprehensively sample Zambezi, this tree would not fit on this page. It would be much, much. So from the tissues that we have, you find that the Lower Zambezi is highly represented with the Ruo, with this Shire one, as well as this one which I'm calling Palombe, as well as even this one, Nyangombe. So these are all from the Lower Shire. Then we have Chiloglanis Zambezi, which is a widely distributed one, as well as another Upper Zambezi one, which is Kabongo here. And even when using species delimitation techniques, you find that they all form unique taxonomic units. So this is the distribution. So for taxa that they have a mixed bag of distribution patterns. And what I mean by that is that intuitively, you would think that highly specialized species would probably be found in very unique habitats. Thus would not necessarily be very widely spread. And you find that a lot where you have species that are restricted to just small river sections, including the ones that we described, you know, Pungwe, the Buzi. But then again, you find this very interesting species, Zambezi, where you find it in the Upper Zambezi, you find it in the Okavango, you find it in the Middle Zambezi, you even find some sections in the, this is still Middle Zambezi, now it's now Lower Zambezi. So this species is found throughout the system, which is very peculiar. Then you even have this one, which I'm calling Kabompo. Again, you find it in the upper sections, as well as in the Kabompo River upper sections, as well as some within the upper sections of Okavango as well. So there is quite an interesting distribution pattern going on there. But again, before we get there, we still need to give these things name, and also we still need to fill in these gaps, because the reason why there are no samples here, it's not because there are no Chiloglanis, it's just simply that no one has collected and lodged samples in accessible collections. So when this gap eventually fills, the story is even going to be much more interesting and more complex. So I just took examples of the genetic distance. So you find within the intraspecific, these are just examples. You find it's very low. But even with this widely distributed, the Zambezi, which at first I was like, no, it's a one species. You see, it's very low intraspecific difference, less than 1%, about 1% within this widely distributed one. Whereas when you compare it to other species, you are talking about 5% all the way up to 10, 12, 15%. So clearly, intraspecific diversity is very high, but within the species, we have very low diversity. Okay. So then we did the morphometric analysis. Then predictably they would group together because they tend to all have this very form meets a function type of body form, whereby they all, because they all generally are found in riffle kind of systems, fast flowing waters, they have kind of evolved into this very efficient form. So the body-wise, the body shape doesn't differ that much, and that's why you find a lot of clustering. But even when you look closely, there are going to be one or two characters that are going to be very unique. So I'm just going to give you a few examples. So, for example, body depth. And this one is a species from Middle Zambezi. You see, it's very deep body, and even there are a lot of characters that you find here, deep body, even when you were to look lengthwise, a really short, rotund type of animal. This is found in the Middle Zambezi. Unfortunately, we actually don't have genetics for this one, but from the morphology itself, it's very clearly distinct from all that we have. This was just comparing it to a more slender form, which is fasciatus. So you see very different body shapes there. Another character that you can look at is the Colobedango depth. Again, very narrow, very deep. And these also, I think these also co-occur. Another character, so these are just a few. Even the length of the spine, you have one from the middle, upper Zambezi, very tiny little spine here, whereas others have much, much longer spines. You look at other characters like the barbels as well, because they have 2 sets of barbels, 3 sets actually. You find that the length of them actually differs depending on the group. So there are quite an assortment of different characters. A character that I love very much because it's very easy to identify is tubercles. So you find some have this ridge-like tubercles, which just like form little ridges. Then you have others which have these more conical, pimply type things going on their skin. So looking at this, you can clearly see there are very clear morphological differences, very clear genetic divergence, as well as even if you want to go to, to the counts, you will find different counts on the number of teeth as well. And the Suckermouth, yeah, when you look at the teeth as well, you'll be surprised sometimes what you see down there. So you have different distribution of the mandibular teeth. Some are widely spaced, some bunched together like this. Some are closely spaced and much longer. And even when you do the counts here, you find different characters. So they may have a very similar body form, which probably attributes the reason why historically they've just been called one thing. Because if you don't really look closely, you would assume it's the same thing. But when you start looking much, much closer, you actually start picking up a lot of very interesting characters. And probably a lot of that has something to do with the ecology, which we'll probably get to once we sort out this big problem. So now just going to the discussion, you find that integration, this integrative taxonomic approach is important, and it has enabled us to identify at least 6 species. But I will guarantee that there's much more diversity beyond the 6 species that we are probably going to describe here. As well as a more wider and comprehensive sampling will probably review a lot more diversity within the Zambezi system. And a lot of these species, unfortunately, are very narrow range species. And you find a lot of high diversity in high altitude areas. For example, the lower Zambezi. This is the Mulanje Massif. It's drained by the Ruo River, which then feeds into the Shire River down there. And within this massive, relatively speaking, you find that there's at least 3 things that are co-occurring. And unfortunately, this is a very high agriculture. There's a lot of agricultural activities happening there because there are a lot of tea farms here, and all of that then has an impact. But right now, we don't know how to assess the impact because, again, this is as it is. They don't have names. They're just those little Suckermouth catfishes in the river. So at least once we have names, and then we can then at least initiate the processes of assigning conservation statuses for all these things. And it also helps out for a lot of these conservation organizations. They need to have formal scientific evidence to then be able to think about even conservation. Because it's difficult to think about conservation when you are unsure of the diversity that's occurring in an area. Similarly, you find, again, that's why they spread the Zambezi one. I mean, this is a very interesting ecological question that I would like to also find out. Like, it's found all the way up in the Zambezi. Like, we're talking of an elevation drop of more than 600 meters. Because in the highlands, the Zambezi, the way it's structured, in the upper Zambezi, you are very high, about 1,000 meters up. By the time you reach the mouth, you're like, you've dropped. And a wide range of environments and characters there, yet you still find the species as widely distributed. So that's a very interesting ecological question that needs to be tackled. Then, of course, some of the threat. So the presence of Chiloglanis species within small ranges adds to the urgent need for conservation, because not only are they found in small areas, also they are found in areas of importance, like these montane areas where a lot of the water that people drink comes from montane areas. We're talking in the Eastern Zimbabwean Highlands. We're talking of the Amatola Highlands as well. All of these montane areas, that's where a lot of the water that we drink comes in. So conservation of these small catfishes, along with other taxa that occur there, has an impact on society, because then it will also help in protecting these these areas of important water resources. So unfortunately, as it is, there is a lot of mining that occurs in all of these areas. So shedding light on the diversity of these species also is important, because it then helps out with the conservation of these areas of important water resources. So I'd like to thank the SAIAB team, as well as all the people that have helped me out. I didn't do all this work by myself, the collections people and SAIAB for its provision, and of course guidance from my host, Prof. Chakona. Thank you.
Thank you very much, Tadiwa, for a very enjoyable talk. Are there any questions for Tadiwa? I hope there are lots of questions for him. Any questions in the room?
Yes, thank you so much, Tadiwa, for your comprehensive talk. And I like the fact that you started from explaining what taxonomy is to most of us who use most of the species that are already identified. So my question is, if a species has been identified, is distributed globally, in the naming process, for those names that we use to honor someone else, can it be applicable also globally? If you name your tilapia Tadiwa, then would it be a global name, or it's a region, like it's in Africa, blah, blah, blah.
Okay.
And also, sorry, another question is the renaming of species. I think previously maybe the species were named or identified based on the morphology. Does it mean now to rename or confirm other species, the DNA process has to be applicable in all the renaming?
Okay.
Thank you.
So to answer your first question, this is one thing that I know Albert always talks about impact, research impact. Once a species has been named formally, that name applies global. So it has a global impact. Whoever is going to work on that group has to use that name. Then that's the taxonomy, the formal taxonomic name, right? But also in addition to that, there are common names as well. That people name that, we call it a hiliminki, we call that, I don't know, Suckermouth catfish. That may be a regional thing. That one depends on where you found the fish, where it's collected, and the people who use that fish. So that one is not strictly as enforced. But the formal taxonomic names, there are actually regulations that enforce that, how the naming process and how if you want to change a name, what's the process. So it's not, it's a very rigid. I think that's one of the things that people complain about taxonomy. It's very rigid. So once it's already in the books, formally described, it's always going to be there, and there are processes that you take. But to answer your question, yes, a formal taxonomic name applies globally. But of course, every species has its own regional name depending on the people that use it. Then the second name is the renaming. So essentially what we're doing, for example, this calendar, it was Newman and I, we are in the process of renaming a lot of these things. So because of the advent of new scientific methods, DNA is a very, very useful tool. But ultimately, it is not the only tool. So if you have other evidence that is not genetic, that is acceptable, you can do that. You can, in this day and age, you can still name a species which just morphology without even any genetics. It's very possible. But again, you have to make a very strong scientific case for that.
Thank you, Tadiwa. I see there's a question online from Dennis. So we take that one. Good luck.
Yeah, hi. Can you hear me?
Yes, Dennis, go ahead.
Yeah, hi. Oh, this is fascinating. It's lovely to see all this work being done. A few questions and comments. All the stuff around melanges, are those the fish that I collected in 2011?
Yes.
Yeah, that's great. There's even more diversity there than I thought. Yeah, in the Shiri, I was there, 2023, in the lower Shiri and middle Shiri. We were getting two very different Chiloglanis then. So the specimens are still in Malawi. Would you like to look at those?
Yes, Dennis, I would appreciate any material that you have. I think maybe you can have a chart after this, then we can kind of...
Yeah, Yeah,
talk about.
okay. And are you in touch with Martin Jenner? Because he had 7 different species of Chiloglanis from Malawi. It'd be very useful to collaborate with him.
Yeah, I think we would be willing to collaborate, but right now from my study, we're still mostly on the Zambezi, but the goal for me at least is essentially to extend it to all those areas, because like you're saying, with the few specimens I've also seen from the Malawi, I haven't really started working on them. I was just browsing through. You can definitely see there is quite a lot of diversity there, but definitely in the future, that's something that I think is a worthwhile pursuit.
Yeah, and I think in the upper Zambezi as well, if you look at the Four Corners report, I think we had about 4 different species of Chiloglanis from there.
Yeah, yeah.
Yeah, we should organize some more expeditions.
Definitely.
Yeah, okay, let's get in touch by email, and I'll sort out photographs as well for you of those fish.
Thank you, Dennis. Yeah, I'll definitely contact you.
Yeah, okay, great.
Cheers.
Thank you very much, Dennis. I see that you have a lot more work to do. Looking forward to that. I see there's another question in the room, in the back there.
Thanks, Tadiwa. I may have missed this in your talk. Just you mentioned something about like the methodology, and you showed a graph there, and there's a lot of overlap. So I'm just interested to know with those morphological overlap. You said there are like a few features that you use to distinguish these clusters. I'm just interested to know with those clusters, those separate clusters, did you look at the genetics as well, and if they correlate the morphological clustering, does it also correlate with the genetic clustering?
Yes, so okay, maybe so each graph there, it represented a genetic lineage. So essentially, you start off with your genetics, then you kind of have an idea of the groupings. Then you go in there looking for morphological characters that can distinguish each group. So you find, of course, there will be a lot of clusters. It's because that was just mostly morphometrics. So, you know, the whole allopatric growth of a fish, it's not, there are obviously going to be some overlaps, but there are some characters that would distinguish one group from another group. One group from another group. So you then have to, especially when you are dealing with so many of these unknowns, you then have to try and find individuals that separate at least from each group. So I only showed a few, but there are quite a lot more, because then would be all day. But yes, each morph is linked to a genetic lineage.
All right, thank you. I see there's an online question from Martin. Then we can come back to a question in the room.
Hi, good afternoon, everyone. Yes. Oh, good morning still. Martin Jordan from Cape Nature. Yeah, Tadiwa, we met back in 2016 when you, I think, were a first-year master's student. So it's really been very good to see your research progressing. So thank you very much for a very interesting talk. I just had a quick question. You mentioned that that bass was quite a problem for the Chiloglanis. Are they able to coexist with invasive bass species to a certain extent, like we've seen with some of the Austroglanis in the Cape do, or would you expect bass to be able to completely extirpate some populations? Thanks.
Thank you, Martin. Yes, wonderful trip. I remember that 2016. Oh yes. Okay, I wouldn't be able to answer that because I've not done much ecology work on the, I mean, there is still quite a lot of things that we don't know ecology-wise. What I can just say is for some of the, particularly the one that we described, Carnatus from the middle Zambezi, it was collected from the size that it was collected, if I remember correctly from the collection, there were also bass there as well. So I don't know if that is a sustainable coexistence or something that we need to look into. But generally, I mean, consensus is we kind of feel like invasives generally have a negative impact. But to what extent, I don't know, and I'm not able to answer that question at the moment.
Cool. Thank you.
Yeah.
Thank you very much. Last question in the room. Is there another question? Going once, going twice, once. All right. Thank you very much, Tadiwa. Well done. and I look forward to your upcoming work with Dennis. So I'll keep an eye out. All right. Thank you everyone for coming through and